welcome back to Inspiring Minds. I am Rishabh, back again with a new video. In this video guys, we will talk about the gluconeogenesis. It is a very new but I think it is a familiar concept to very much of you. In the last videos we have discussed about the glycolysis. The glycolysis in which the glucose is broken down into the pyruvate. Okay, so this process was called as glycolysis. But in this video, what we are talking about is gluconeogenesis. What it means is glucose means gluco, neo I will tell you, and genesis you all know that it is synthesis. It means the synthesis of glucose. But from the neo means new resources, means how the glucose is formed. Tell me, glucose का primary source क्या है? Carbohydrates. Okay. But this is an old or a traditional source. I am talking about the new source. Means the formation of the glucose from the new sources other than the carbohydrates. And what are these sources? Proteins and fats. So simple definition is gluconeogenesis means the synthesis of glucose from the non-carbohydrate resources. Okay. So let's discuss. And what we will do is we have to form this glucose here. And from what? From obviously the pyruvate. Means we have to just reverse the glycolysis process. So sounding familiar and sounding very easy. आपको ऐसा लगेगा कि बहुत आसान है. Just pyruvate से back उल्टे चले जाओ और सारी glycolysis की process को reverse कर दो. लेकिन friends, let me remind you, in the glycolysis I have told you, if you have watched my video, in the glycolysis there were one rate limiting enzyme which name was phosphofructokinase. But major importantly, there were 3 irreversible steps in the glycolysis. So they are irreversible and we cannot reverse them. So for that reversal, for that irreversible steps, we have to find an alternate way to produce the glucose from the pyruvate. So see, what we have to do, we have to just convert this pyruvate into the glucose. लेकिन एक drawback है, it is not the exactly opposite of glucose. It cannot be the opposite of glycolysis. Why? Because there are 3 enzymes which catalyze the 3 irreversible reactions in the glycolysis. We will needing some special enzymes which will reverse that reactions, irreversible reactions in the glycolysis. So let's see what are those enzymes and what are these reactions. See, firstly in the glycolysis, one enzyme was pyruvate kinase. You can watch this video twice. I will firstly I must tell you that gluconeogenesis is a very complex and very difficult topic to understand. It took me around 3 days to understand this topic, and now I am here to present to you in a similar way, in a fashionable way, so that I can make it easy for you. Okay, so you just open the glycolysis in front of you and then refer this and watch this video. Okay, so glycolysis we use the pyruvate kinase. In the gluconeogenesis, in place of pyruvate kinase, to reverse this enzyme's reaction, we will use 2 enzymes. One will be the pyruvate carboxylase and other will be the PepCK. Okay. And also to remind you, the PepCK enzyme was also used in what? In the glycolysis, but that was PepCK1 and here we are using the PepCK2. Okay. Now, PFK1 we used. Then in place of PFK1, what we will use? The fructose 1,6-bisphosphatase in the gluconeogenesis. And the third enzyme what we used is hexo or glucokinase, okay, which converted the glucose into glucose 6-phosphate in the first step and used the ATP. Okay, and these 3 are the irreversible steps of the glycolysis, and the enzyme which we will use in place of hexokinase will be the glucose 6-phosphatase. Okay, so just similarly, in glycolysis we had 3 irreversible reactions which uses the 3 different enzymes, and in place of that we will use 4 enzymes in the gluconeogenesis. 4 enzymes in gluconeogenesis in exchange of 3 in the glycolysis. Okay, now moving forward, there is a rule of 2 in the gluconeogenesis to make it easy. It is just a simpler process, not difficult. Rule of 2, and how many 2's? 3 2's. Okay, so you can remember as 3 into 2, like 3 into 2. Okay, so let's see. There are 2 compartments, means the 2 places where the gluconeogenesis will occur. First, it will start in the mitochondria, ends in the cytoplasm, and then also it goes in the endoplasmic reticulum also, but for a small period of time I will tell you. Then there are 2 organs. Gluconeogenesis in our body occurs only in 2 places. Liver, majorly liver. You have to remember this. Liver is the main organ for gluconeogenesis, and a little bit occurs in the kidney. Okay, And gluconeogenesis की जरूरत क्या है? Why the gluconeogenesis is occurring in our body? If we are eating the carbohydrates, we are getting the glucose. So why there a need that carbohydrate is absent in our body, so that we can use the protein or the fat which we are eating or which is deposited in our body to use as a glucose source. So two situations will be the fasting or starvation condition, in which we are not eating the food, and if we are not eating the food, we are not getting the carbohydrates, hence we need some other alternate source of the glucose. Okay. Or in the case of diabetes, the patients of diabetes which has insulin deficiency, and their blood glucose level is already lowered, and they need instant, or I should say regular uptake of the glucose, which they can get from the other sources. Okay. So now let's compare what happens. Like I can compare the diabetes also as a fasting condition only, because the glucose is deficient in the blood. So what happens? Let's compare the two conditions. The first condition is the feeding condition, and other is the fasting or starvation. Okay. Now first let's consider about the feeding condition. What happens is, we eat the food, the insulin level rise up from the pancreas, the insulin will released, and it will start the link reaction. You should remember that the link reaction is initiated by the insulin, and the insulin, if deficient, the link reaction will also be stopped working. Okay. So what will happen is, what link reaction does is, link reaction joins the glycolysis with the TCA. The last product of the glycolysis is pyruvate, and the first product of the TCA is what? Acetyl coenzyme A. So this pyruvate will be converted into acetyl coenzyme A, and this acetyl CoA goes into the TCA, okay, by the enzyme this pyruvate dehydrogenase, and this is the link reaction. Okay. Now this TCA will occur. What happens in the fasting condition? If we are not eating the food, no carbohydrates, hence we will have to break the fatty acids, the beta oxidation of fatty acids, and beta oxidation of fatty acids also give the acetyl CoA. So see, we had the pyruvate, we had the glycolysis. Okay, glycolysis occurred, glucose is broken down into the pyruvate, but this link reaction will not be occur, because we are in the fasting condition or a diabetic patient in which insulin is not there, then link reaction will not be there. But we will break the fats, and we will get the acetyl CoA. This acetyl CoA will go into the TCA. Okay, so let's see now. This pyruvate through the phosphodehydrogenase enzyme will convert into the acetyl CoA in all the patients. But due to beta oxidation of fatty acids, acetyl CoA is increased. Suppose in these patients, with the normal eating patients, the acetyl CoA has a value of X. Okay, has a value of X. But in the beta oxidation of fatty acids, more X. So in the diabetic patients or the fasting condition patients, how many acetyl CoA? 2X. It means the acetyl CoA enzyme will be increased than the normal. Its value will be increased than the normal. And this will get the feedback inhibition to this PDH enzyme, and this conversion will be stopped. The pyruvate will not get converted into acetyl CoA, because this मैं कह सकता हूँ कि acetyl CoA की कमी जो हो रही है पूरी, वो कौन कर रहा है? This beta oxidation of fatty acids. Okay, so अब क्या होगा? This pyruvate also will get accumulated. See, this pyruvate will get accumulate and then goes into the gluconeogenesis. What we said in the initial part, that this pyruvate will then reversed as a glucose. This is called the gluconeogenesis. Okay. बिकॉज हम इस बीटा ऑक्सीडेशन ऑफ फैटी एसिड्स को ज़्यादा टाइम तक कंटिन्यू नहीं कर सकते, बिकॉज इट इज़ अ एक्सपेंडिचर प्रोसेस, वेरी एक्सपेंसिव. ओके, सो इन होल एनर्जेटिक्स during the fasting or starvation, this link reaction will be switched off, okay, because of insulin deficiency, and rest all will work. Like this acetyl CoA will go into the TCA, then the ETC, and initially the glycolysis is also occurring. So everything is going on, but this link reaction will be stopped. Okay, now let's see what will happen. Now we have the pyruvate which will go into the gluconeogenesis. How this process will work? Let's see this. This pyruvate, which is a 3-carbon compound, will be initially added by this enzyme pyruvate carboxylase, and the CO2 will also be added. This acetyl CoA, which was the TCA intermediate, which will go into the TCA for the first step of this is the first step of gluconeogenesis. For the first step comes here and will activate this first step and will convert this into oxaloacetate. This is a 4-carbon compound. So pyruvate will be added by the CO2, 1 carbon, and this oxaloacetate will be formed, which is the 4-carbon compound. Okay, so this acetyl CoA during the fasting condition, see, does 2 works. What? Firstly, it will inhibit this PDH complex to stop the conversion of pyruvate into the acetyl CoA. Also, it will, I should say, what is it doing there? See, stimulating. It will stimulate the first step of gluconeogenesis also. Okay, and after that the remaining acetyl-CoA will go into the TCA. Okay, similar. Now, in place of 3 irreversible reactions in the glycolysis, what we have used? We have used actually 4 enzymes for the gluconeogenesis. Okay, now see how this gluconeogenesis process works. It is just now you know the basics. I just have to formulate you in the step-by-step process. It will be very easy. So I will here tell you. Now I should first tell you the 2 conditions for which the gluconeogenesis occur are the diabetes or the fasting condition. You got it. Now the 2 organs, liver and the kidney, you got it. But you have been in a little dilemma, like how the 2 compartments, because the glycolysis what occurring in the cytoplasm, TCA occurring in the mitochondria, everything else in the mitochondria, some in the cytoplasm, but gluconeogenesis is occurring in both. So how this is possible? So let me formulate this in a flow chart and will tell you part by part. Okay, see, this glucose by the glycolysis converted into the pyruvate. Simple process. The pyruvate will convert into acetyl-CoA unless the beta oxidation of fatty acids will occur. This acetyl-CoA will increase, give the feedback inhibition to the PDH, and this conversion will be stopped. Okay, this pyruvate will be accumulated, and now the process starts. This pyruvate will be accumulated in the cytoplasm, but the excess pyruvate cannot be handled in the cytoplasm. Hence, this pyruvate will go where? This pyruvate will go inside this mitochondria. Okay, now comes the role of mitochondria. I told you that gluconeogenesis begins where? In the mitochondria. Here, by this pyruvate, excess pyruvate into cytoplasm goes into the mitochondria. This pyruvate, now first see the enzymes which were using in the gluconeogenesis. See, just learn these quickly. What are these enzymes? Pyruvate carboxylase, number 1. Number 2, PepCK. Number 3, fructose 1,6 bisphosphatase. And number 4, glucose 6 phosphatase. I will tell you compartment by compartment because you can be asked by the questions, where is this enzyme used? Or gluconeogenesis के अंदर mitochondria में कौन सा enzyme use होता है? You can be asked questions either way. So you should be prepared every way. Okay. So let's see. This pyruvate will be converted by the pyruvate carboxylase. You know the first enzyme? Okay. This pyruvate will be converted into the oxaloacetate and this oxaloacetate into the malate. But the point to be noted is pyruvate carboxylase will use what? ATP. So the gluconeogenesis is a expensive process, I told you. Okay. Now this malate is in the mitochondria through the shunts. Okay, through the shunts. This malate will be coming outside of the mitochondria. Now you must be remembering that shunt was there, but this is a wrong concept. I told you the shunts are only for conversion or sending from cytoplasm to mitochondria, not from this way. Okay, if that substance is coming from mitochondria to cytoplasm, there will be no shunt used. Okay, this malate just directly will come outside into the cytoplasm. This malate again converts into oxaloacetate and the second enzyme PepCK. This PepCK will be again used and GTP also used. So one GTP and one ATP has been used. This oxaloacetate will be converted into the PEP. Now the third enzyme in the gluconeogenesis, PG kinase. Okay, this will also use the ATP and will convert into the fructose 1,6 bisphosphatase. Okay, now this fructose 1,6 bisphosphate will be converted into the fructose 6-phosphate. Okay, now through this enzyme, and the enzyme will be glyceraldehyde 3-phosphodehydrogenase. You remember in the glycolysis also, if this was the step from fructose 6-phosphate to fructose 1,6 bisphosphate, this only enzyme was used. And you know the important point, this enzyme is only capable of using the NADH into the cytoplasm. This NADH present in the cytoplasm can only be used by one enzyme, glyceraldehyde 3-phosphodehydrogenase. Okay, now this fructose 6-phosphate will be converted into the glucose 6-phosphate, and the fourth enzyme used will be the glucose 6-phosphatase, which will be converting into the glucose. So this will be the whole process coming in a fashionable way where you can appreciate how the 3 compartments, mitochondria, then cytoplasm, and then ER. What are the 3? Firstly, the mitochondria, then comes the role of cytoplasm, and then the role of ER. It will come into a play role, and then this pyruvate will be converted into the glucose to complete the process of gluconeogenesis. So let's just quickly formulate again in the mitochondria. What will happen in the mitochondria is the pyruvate will be converted into the malate and oxaloacetate, and this oxaloacetate into the malate. This was occurring in the mitochondria. Then we reach where? In the cytoplasm, and a whole bunch of reactions will be occurring in the cytoplasm. Now also let me tell you by side in the mitochondria which enzyme was used. Only one enzyme, which was the pyruvate carboxylase. In mitochondria, enzyme will be pyruvate carboxylase. Okay, now in the cytoplasm other two enzymes will be used. What were they? The pep CK and what? Fructose 1,6-bis phosphatase. These two enzymes will be used in the cytoplasm, and where will the reaction left? The reaction will be at the end. This malate again converted into oxaloacetate, then the whole bunch of reactions you can see there. This pep into the fructose 1,6-bis phosphate, fructose 6-phosphate, and glucose 6-phosphate. So the end product will be, in cytoplasm I'm talking about, is glucose 6-phosphate. Okay, now the last, this glucose 6-phosphate, this is a very interesting thing. This will come into the endoplasmic reticulum. And why it will come into the endoplasmic reticulum, let me explain you. In the endoplasmic reticulum, we have an enzyme, glucose 6-phosphatase. You know, the number 4th enzyme of the gluconeogenesis, and this will convert this glucose 6-phosphate into the glucose. So where we will get this glucose? We will get this glucose into where? Where? Tell. It is in the ER. Very good. Okay. So why we get this endoplasmic reticulum into the role? Because we have this enzyme only present in the endoplasmic reticulum. Why? क्या ये एंजाइम साइटोप्लाज्म में काम नहीं कर सकता? बिल्कुल कर सकता है, लेकिन हमने इसको एंडोप्लाज्मिक रेटिकुलम फिर भी रखा. सी वाई? बिकॉज इफ इट ग्लूकोज 6-फॉस्फेटेज एंजाइम साइटोप्लाज्म के अंदर होता तो ये इस रिएक्शन को साइटोप्लाज्म में कर देता, और ये ग्लूकोज हमें कहाँ मिलता? साइटोप्लाज्म में, और साइटोप्लाज्म से कहाँ चला जाता? तुरंत ब्लड में चला जाता, एंड इट विल एस्केप इनटू द ब्लड एंड विल बी यूज्ड एज द एनर्जी सोर्स. लेकिन हमें अभी क्या करना है? हमें इस ग्लूकोज को बचा के रखना है, ताकि जब हम फास्टिंग स्टार्वेशन कंडीशन से निकलें या जब हम उसके एंडिंग वर्ज पे हों तब इस ग्लूकोज को हम यूज कर सकें. इसलिए हमने ये एंडोप्लाज्मिक रेटिकुलम की हेल्प ली. इस एंजाइम को हमने कहाँ रखा? ग्लूकोज 6-फॉस्फेटेज को एंडोप्लाज्मिक रेटिकुलम में. ये रिएक्शन कहाँ कराई? ईआर में. और ये जो ग्लूकोज होगा वो स्टोर कहाँ होगा? ईआर में. ओके, एंड जब इसकी नीड आएगी, व्हेन इट विल बी नीडेड, व्हेन दिस ग्लूकोज विल बी needed, we will use this glucose, we will bring that glucose from ER to where? To the cytoplasm, and from cytoplasm it will go into the blood and will be utilized. So for this, because you know glucose has a tendency, it cannot stay in a cytoplasm for a long time, it can escape out very quickly. So for preventing that escape, we have to use this endoplasmic reticulum, but for a very few time. That's why if a question is asked, where is gluconeogenesis occurring, and you have options like your A option is mitochondria, cytoplasm, ER, and other option is mitochondria and cytoplasm. You should remember and tick this first option, because ER has a role, a little role, but yes, a very crucial role to enable the working, a proper working of glucose 6-phosphatase. Okay, so this is a part 1 of the gluconeogenesis, in which I have told you about the process, about the enzymes, about the location, and about the organelles where this process is all occurring. It is a typical process, basic process which always occurs if you have heard of the intermittent fasting, and any fast, any starvation, और जिसे आप व्रत रखते हैं, किसी भी फास्टिंग कंडीशन के अंदर ग्लूकोनेोजेनेसिस आपकी बॉडी में होती है, रेगुलर बेसिस पे होती है. रात को भी आपकी बॉडी में कई बार ग्लूकोनेोजेनेसिस हो जाता है अगर आपने डिनर प्रॉपर्ली नहीं किया या फिर आप सुबह उठ के ब्रेकफास्ट टाइम पे नहीं कर रहे हैं. सो यू शुड नो अबाउट दिस, एंड लेट मी टेल यू, इट इज़ अ वेरी हाई यील्ड कॉन्सेप्ट इन एवरी एग्जाम, बिकॉज अ गुड questions are asked about this, so you should be remembering all of this. But there are a few things left about the substrate, like I told you, the fats and the proteins can be used as a glucose source other than the carbohydrates. So which amino acids, which proteins, which fatty acids are used, because all fats or all proteins cannot be used as a substrates. So what are those? What are the energetics? क्या इसके अंदर बहुत ज़्यादा एनर्जी लॉस हुआ? सी, क्योंकि इसके अंदर कितना ATP हमारा खर्च हो गया. इन द फर्स्ट स्टेप पहला ATP खर्च हुआ, सेकेंड GTP खर्च हुआ, और तीसरे ने भी ATP यूज़ किया. सिर्फ एक जो फ्री का एंजाइम था जिसने फ्री में काम किया, दैट वाज द ग्लूकोज 6-फॉस्फेटेज, लेकिन एक्चुअली फ्री नहीं है, इसने भी काम किया ER में, जिसके लिए और एनर्जी वेस्ट हुई. तो यहाँ तो अभी दिख रहा है कि बहुत एक्सपेंसिव हो गया. तो कितनी इसका यील्ड है, कितना इसके अंदर एनर्जेटिक्स है, वो सब हम बात करेंगे इन द नेक्स्ट वीडियो. सो जस्ट स्टे ट्यून्ड फॉर द नेक्स्ट वीडियो एंड टिल देन revise this concept, learn the gluconeogenesis, but first revise the glycolysis, and then if you will study this, it will be very easy for you. I hope I made this complex a difficult topic for you simple and easy. So just enjoy this lectures and see you in the next one with a gluconeogenesis part 2, in which we will talk about the energetics and the substrate for the